Thanks so much, Jackie. It's really a pleasure to be here, and I'm going to give you a little bit of a perspective from my point of view. So, as Jackie said, I'm a microbial ecologist, and rather than thinking about microbes in the human body, I think about how microbes are interacting in the environment to ultimately create an environment that is habitable for us. And I wanted to start with just maybe this is not working. Do we know? Maybe I'll do it here. Okay. All right. By a show of hands, how many of you have ever been out stargazing? All right. That's what I figured, right? Away from like the city light pollution and all of that, right? So I want you to tap into that sense of awe and wonder when you're looking up at the night sky, at what seems to be like an infinite number of celestial bodies out there, right? Remember that feeling. That is the same feeling that I have when I look under the microscope at the millions, sometimes billions of microorganisms that can be in a dropper full of seawater or a little pinch of soil or sediment. What I'm showing you in the background here is not celestial bodies, but rather these are microorganisms that I collected from a seawater sample and pulled it down, maybe just a few milliliters. The larger green spots you see here are the bacteria and the viruses, the bacteria and the archaea that make up some of the microorganisms, and the really small pinpricks of light, those are environmental viruses. They are so infinite, in fact, that in the ocean alone, there are orders of magnitude more microorganisms than there are estimated stars in the universe. So it's profound when we think about it. Now, this microbial life actually translates to a significant fraction of the total biomass in the oceans, right? These things are incredibly small. We can't see them with our naked eye, yet they represent 70% of the total biomass in the ocean. It's not the big charismatic megafauna that we like to think about when we think about the oceans. It's really, that's a small sliver compared to this microbial life that's all around us, but oftentimes we don't think about it because we can't see it. Now, as Margaret said in her lovely talk just before the break, this microbial world is incredibly diverse. And what I'm showing you here, you can think of this sort of as a family tree. This is like Ancestry.com going back, maybe almost 4 billion years. And it's a prune tree, but it's showing you the major domains of life as we know them today. The bacteria, the archaea, and the eukaryotes. And the majority of this diversity, even within the eukaryotes, is microbial, right? That is single-celled organisms, not these big things. So to put this into context, you sit here at the tip of this branch, and in this tree, corn is your nearest cousin, right? It's a huge, huge amount of diversity. The other really key point I want to make is that the majority of the trees on this branch of Tree of Life represent uncultured microorganisms in the environment. That is, organisms that we really only know from their DNA sequences. So Margaret made a really great point in that we have opened up new vistas through the ability to sequence deeply in the environment. And what that has shown us is there's an incredible amount of diversity there, but we don't understand them at the same level as we do growing an E. coli on a plate. Right? So to me, as a microbial ecologist, it's an incredibly exciting and challenging opportunity to really start to put these organisms in the context of what are they doing in the environment. And they are playing really important roles in how the biosphere operates, right? These microorganisms are responsible from doing things like pulling the inert dinitrogen gas out of the atmosphere and turning it into a bioavailable form for all of life on the planet. The oceans, microbes in the oceans are pulling CO2 out of the atmosphere and sequestering it, helping to control our climate. And we have microorganisms that are also consuming and producing other types of greenhouse gases like methane. All of these things are going on all around us, and even though we don't think about them all the time, they're creating environments that are really necessary for us to live. And they've been doing this for billions of years. They have totally shaped the landscape of our planet over time. Now, I wanted to share one sort of example that hopefully will resonate with you, and that is oxygen. We all need it, right? And what I'm showing you here in this movie are microorganisms in the ocean that, like a plant, use light energy to fix carbon dioxide and in turn split water and produce oxygen. Okay? So the oceans are literally breathing for us here. And what we can see is this is a satellite image detecting a pigment in those organisms that's used for light harvesting. This is chlorophyll in these organisms, and you can see how dynamic they are over space and time. So this is like a year in the life of life in the ocean, right? So in the time that I've been up here speaking, half of the oxygen that you have consumed has come from these little organisms in the ocean. It's pretty profound. Okay. So we've learned quite a bit about microbes in the surface ocean, aided by things like satellites and being able to analyze the surface environment, but most of our watery planet is far more remote. The majority of water in our ocean is in deep conditions, and in fact, something between 5 and 10% of the seafloor alone has been mapped at any sort of sufficient resolution for us to be able to understand it. Far less of that environment has been actually studied by scientists. This is really one of the last greatest frontiers we have on our own planet. And there are critical processes that are going on within this ocean world, just like in the surface. And one of those is related to methane. So the oceans actually are the third largest potential source of methane on our planet, and a lot of this is locked up in ice along the continental margins here. Yet, when we look at the amount of methane in the atmosphere today, even though it's been increasing over time, the oceans actually don't contribute very much to the atmosphere. And that has everything to do with the microorganisms that are living in these deep environments that are consuming it. Somewhere around 80% of that methane is consumed before it can ever get into the atmosphere. And without that ecosystem service, we would be in big trouble. So the challenge for me is, how do we study something that is a micron in size that's living over a mile deep in the ocean? And so for our work, we get to use submersibles that allow us to go to places where methane is venting to the seafloor. And in these places, there are tremendous amounts of symbiotic associations that have developed, all based on chemical energy. And this includes animals that have teamed up with microbes as well as the microbes themselves. This is a great natural laboratory to study methane cycling. Within every square centimeter of soil, we have billions of organisms, of which 80% of that biomass are from organisms that are consuming methane. I'm showing you some fluorescent images of some of these microbes that we've been studying here in this image. And so you can see in that image that these organisms not only are abundant in the environment, but actually in some cases they're physically associated with one another, which suggests something about their types of interactions. So the challenge both for my work and for the greater microbial ecology field, is how do we start to figure out what are these diverse microorganisms are actually doing in the environment? I'm interested in methane, but there's a lot of other processes going on as well. And can we start to understand not only the diversity, but how they're metabolically interacting with one another? And so in my research, we have used not only the sequencing data, the genomic information that is encoded in all of the cells in the environment, but also the chemical information that's found in those cells as well. Each of these things used together can give us greater insight into what these organisms are doing and what they're eating in the environment. And so by pulling in tools from geochemistry, we're getting sort of a more holistic picture of how these organisms are connected to the environment. And one way that we do this is through the use of stable isotopes. And for those of you who this might be sort of a new concept for you, many elements exist in different forms in nature. Their atoms exist in different forms, namely in the number of neutrons that each possess. And so in the case of carbon that I'm showing you here, carbon-12 has 6 protons and 6 neutrons, and it makes up the majority of carbon that we naturally find in nature. But there's a small percentage, a little over 1%, that it has an additional neutron here, right? Which leads to a very small change in mass. And it turns out that biology can react to these things in very different ways. And so I like to use an analogy that my dear colleague at Caltech talks about in terms of how to explain this. And he uses baseball. So this is Alex Sessions. So you can think of this carbon-12 and this carbon-13 as 2 baseballs, right? They have the same shape and the same size, but perhaps the carbon-12 version of the baseball is filled with cork, and the other one is filled with lead, right? So that cork ball is going to fly faster and further than the lead ball, right? So biology senses those differences. I mean, that's a pretty extreme. It's not quite that extreme, but you get the point, right? So it turns out enzymes also have different ways that they are interacting with these molecules, and this leads to different ratios of carbon-12 and carbon-13 in the biomass of cells that we can then interpret to give us a sense of what organisms are eating. So a classic example in trophic ecology can be found in the plant world, looking at corn here, which is a C4 plant. How I'm orienting you here, this is carbon isotope ratio, right? That's carbon-13 over carbon-12, and we do it in delta notation. You don't have to worry so much about that, but all you need to know is that on the right side here, there are more carbon-13s, and as you go in the more negative version here, there are more carbon-12s, right? And so corn, as a C4 plant, has more carbon-13 relative to, in the ratio form, than something like rice, which is a C3 plant, right? So you are what you eat, right? So if your diet is more rice, your isotopes are going to be more aligned here, versus more corn, which is a lot of the US, unfortunately, which is going to be found over here. So we can put this to the test, and we did put this to the test with my lab by taking fingernail clippings from everybody, and that's all of the data that's shown in these sort of blue dots here. This is the same carbon isotope ratio I was just showing you over here, corn over here, rice over here, and then I've added another dimension here. This is nitrogen isotopes here. And this gives you an idea about trophic structures. So the lower the nitrogen isotopes, the more sort of vegetable-based your diet is, and the further up you go, you get more carnivorous. And we see we have a pretty reasonable spread amongst the members of the lab, and I also had them self-report of whether or not they thought they were a vegetarian or a vegan versus a carnivorous person, and some, you could kind of tell, the proof was in the pudding. Their values were trending a little more meat-like than what they were actually reporting there, right? So anyways, we can use the same kind of concept, but now shrinking it down to the small scale to look at the microorganisms themselves. And when we're thinking about trying to track the process of methane consumption in the environment, it turns out that carbon isotopes are really amazing tracers through their lens of stable isotopes. And that is because methane tends to have a whole lot of C12 in it relative to C13, giving it a very negative value. It's just naturally in the environment. And so we can kind of think, I like to think of this sort of as like the garlic of the carbon isotope world. Like, you just can't mistake it if you've eaten this, right? And so the challenge for us is, how do we start to look for these kinds of signatures in microorganisms themselves? And this has really been a big passion of mine, is trying to develop tools that allow us to do this. And one way to do this is to use a mass spectrometer that allows us to measure things down at the submicron scale. And so we can go out into the environment and find these organisms, take advantage of DNA sequences to fluorescently label specific groups of organisms, and then measure their isotopes, going cell by cell and getting information on that. And when we take a little bit of the sediment from these areas that I showed you from this methane venting environments, what we find is that all of these clusters of cells that consist of archaea and bacteria that are living together in this environment have carbon isotopes that are very similar to what we would expect for methane. So this gave us sort of a direct link that these organisms were indeed assimilating that methane into their biomass and were connected to this process. So we have been interested in studying this group of organisms from all sorts of different angles for several decades now. This has been a passion project of mine. And now the big question is, how do we start to understand what exactly they're doing? We know they're eating methane, but we also see that these two organisms physically or intimately are connected to one another, but yet we don't understand how they're sharing the energy of consuming methane. And how are they doing this without oxygen? There's not a lot of energy to be had, so these guys are breathing sulfate in seawater, not oxygen, which gives them just barely enough energy for one organism, let alone two. So this has been really a fascinating type of symbiosis that we've been very interested in studying. We can use isotopes in different ways now, adding them in their heavy labeled form into the environment to start to ask how those different cells are responding to one another and what is their activity in relation to their nearest microbial buddy, right? And so you can basically create a network of distance of every cell to every other cell. And we can then combine these fluorescent images that are based on the DNA staining of these organisms. In this case, these are the archaea, and these are the bacteria. And we can, by adding this isotope and doing the ratio measurement, we can see where activity is happening in individual consortia. And so what we find is that there is a distance relationship. So the cells you see here on the border tend to have higher levels of activity, which suggests that they're interacting, they're sort of benefiting from each other in that physical distance from each other. So we've been able to use this now, along with the genomic data, to piece together a little bit more detail about how these organisms are operating. And it turns out that these symbioses are doing something really unusual. So they are taking the energy from methane, they're converting it to CO2, and there's about 8 electron equivalents that are generated from this. These organisms are not creating a diffusible chemical that's being taken up and eaten by the other organism. Rather, they are passing electrons directly outside of their cells through a conductive network, and then those electrons are taken up by its bacterial partner and used to drive the reduction of sulfate to hydrogen sulfide. So they're like little energetic powerhouses here. And we would not have been able to figure this out were it not for the ability of pulling information from their genomes and then also using these kinds of isotope techniques and modeling to better understand. These are organisms that live in the deep ocean We can't grow them in the lab in the same way, but yet we can still learn fundamental things about what they're doing. And this gives us a lot of hope for trying to tackle a lot of the other processes that are going on in nature. So I wanted to end again with this concept of microbial life support. I gave you a couple of different examples. One, of course, of the oxygen that we're breathing coming from cyanobacteria in the ocean, and the other with methane oxidation that's happening deep in the ocean through symbiosis. But these are only two examples of a much broader spectrum of incredible microbial metabolic feats that have evolved over the near 4 billion years that microorganisms have been on this planet. And we really can gain a lot of inspiration from learning about how they've figured out how to do all of these incredible ways. Every possible form of way of getting energy from converting matter, some microorganism has figured out a way to do it. Now, this may seem incredibly sort of perplexing to try to get your head around this incredible diversity, but it turns out there are also some common principles that we can start to pull out of thinking about how microorganisms operate. And it's through those commonalities that we really can potentially start to harness the power of microorganisms in a way that betters the planet for us and makes human populations sustainable going forward. So with that, I thank you and take any questions.
Super. Questions? I think there's one right over there, Nora.
I'm Rowena Matthews from Tucson, Arizona. I wanted to ask you whether the ocean acidification that's associated with climate change is likely to severely impact the kind of metabolism you've been describing.
Yeah, it's a very poignant and timely question. For methane oxidation in the deep ocean, it's less likely that this has an effect, but certainly for a lot of the processes in the surface ocean, it is a huge concern. Directly for organisms that calcify, that make calcium carbonate or limestone shells, right? Those will dissolve with increasing pH, and so and the corals are another big issue. But there are other potential ecosystem consequences that we're just really starting to unpack now. So definitely in the surface ocean, that's like the starting point of pulling CO2 down and sequestering it in the deep, and so I think we don't have a good handle yet on ultimately what's going to happen with prolonged acidification.
Hi. David Oxtoby, Cambridge, Massachusetts. You've talked a wonderful talk, really learned a huge amount about it. And my question is, you've of course focused on the ocean, which is a huge topic, but microbes on Earth in soils, is this a completely separate world? Are there any microbes that move from one to the other? And how does soil fit in? And could we learn, is there cross interaction between those?
Yeah, did Diane plant you there for her? No, yeah, so Diane can talk definitely more about the soil realm. It's a good question. So in doing these large global surveys of microbial diversity, actually we see distinct diversity in ocean environments relative to what we see on land. Of course, as you imagine, when you start to get into coastal regions, then there's some crossover between the two. But some of the properties of these organisms and the way that their metabolism is influencing a waterlogged soil versus what we see on a particle sinking through the ocean, from the lens of metabolism, there are a lot of similarities, and so studying one can give us insights into the other. Yeah.
Linda.
Hi, Linda Greenhouse, New Haven. The slide with the biome of the members of your lab was very suggestive. So obviously we all know that there's a relationship between diet and health, and it's better to eat tofu than eat Doritos. But I wonder if anybody's working now on the more granular level that slide suggests, of really looking at the relationship between particular diets in the biome level and human health.
Yes, I think that is a big thing. I think there's lots of companies actually that are making quite a mint on trying to provide you with some insights between your diet and your human microbiome. Absolutely, everything that you eat is ultimately processed by the organisms in your gut, and the specific complement of those organisms and their ability and their efficiency to convert things and the types of metabolites they're producing ultimately is having an impact on all other systems in our body. That is definitely true. Margaret can probably speak a little bit more authoritatively about this topic than I can, but it has given me pause. I certainly pay much more attention to things that I'm putting in my body, and I'm always thinking about my microbes. So.
I'm glad somebody is. I think with that, we should go on to the next.